frente a enfermedades fúngicas emergentes, así como también a enfermedades que se han vuelto resistentes a fungicidas actualmente en uso. La tercera y última ventaja es la inocuidad, pues el ARN de interferencia se degrada razonablemente rápido en el ambiente debido a su inherente inestabilidad química y la presencia ubicua de nucleasas en la comunidad bioecológica.
Okay, now we can start the clock and time for questions. Nanine and then Ellen.
All right, thanks. Yeah, so I really enjoyed your project. I liked how applicable it was to your region, and you had a very detailed plasmid design as well as modeling. And during your HP section, you mentioned something about HP being a two-way street, right? So thinking like a child to remove limits, I kind of like that part. You had proposed on your Joggle site something about injecting the RNA directly into trunks, right? So that seems like it would suggest a lot of manual labor involved. And I was wondering if you did any type of economic modeling there to see what the cost benefit might be and how that would compare to current solutions on the market. You can probably feel free to type your answer in the chat as well if that's more convenient.
You asking about what is the price compared against other products that are current in the market, right?
Yes, specifically with regards to application. So how we're applying this to plants. So does someone have to go to every single plant and inject this, or can we spray this, or?
It's supposed that inject in each plant. Yes. Okay. Because it's trunk injection.
Okay. So how often would someone have to go do this? Every week, every month, or how many times do you think we need to do this?
Because we are using nanoclay, there is a time, for example, this can the during of the effective of this delivery method. I don't remember right now, but yeah, I don't know if other of my team remember how is the time that is in the plant that is effective.
No, sure. That's just something to keep in mind. So if we're going to pitch this to farmers, right, they want to see that it's convenient, easy to use. I can easily swap this out for what I'm using right now, right? So just something to consider in the future. All right, thank you.
Ellen?
Hi. Very nice presentation, nice video. Can you tell me a little bit more about these clay nanoparticle stabilization? How does it work exactly? What is the molecular basis for the clay nanoparticle stabilizing the RNA? You are muted. Are we having trouble accessing other team members? Is that what's going on?
Yeah. We are talking because the person who explained better in this part of bioclinics can't enter, but I don't know if anyone of the group can answer.
Do your best. Do your best. Or maybe let's just go on to the next question.
Yeah, please, and we will try to answer later.
If staff is okay with this, I will continue with the next question. First of all, congratulations, team. Avocado production is really relevant in the world, especially during Super Bowl. So everyone loves avocado, and this is a really important project. What I'm concerned about, or my question is, how do you integrate RNA productions in the parameter of your kinetic models? Where can we see the mathematical expression and equations that can describe this? Because for me, it was not self-evident. Does it make sense to produce 10 grams per liter of RNA when your model biomass concentration is less than 2.5 grams per liter and your substrate is around the same concentration as the production that you're expecting? Please, if the question was not clear, I can develop on that.
Yes, please, can you repeat it in other words?
Yeah, sure. So my question is, how do you integrate the interference RNA production, which is the product that you're interested in making with your bacteria, into your kinetic models? Because I saw that you did some modeling and prediction of how it will behave in a bioreactor. And you have some graphs where you plotted the predicted mRNA production. And I saw that these graphs were like 10 grams per liter of RNA. And in other graphs, you have 2.5 grams per liter of biomass. So does this make sense? How can we see the actual parameters for RNA production in your kinetic models?
Is anyone from your team that was involved in the bioinformatic calculations online now?
I'm sorry, the person who do the mathematical calculation is not here.
Okay, then maybe we can go to the next question. Thank you.
Okay, I guess I could ask a more simple question. So I like the project. I really love avocados also, as a true Mexican. So this is something important, I guess, for our country. But my question is, did you, before, when you were in the design process of your project, did you explore other types of, biological solutions that could be used to kill this pathogen? And if so, why do you ultimately decide on using, RNAi if there are maybe other things that you could use? but I mean other biological solutions, not chemical pesticides, but other stuff, commonly used, other biopesticides.
Yes, when we go to a fundo in Chincha, we see that they use like how to say this, ¿cómo se llama? Me olvidé. What is the name that they use, Rosita, that have like a lot of amount? You remember that they have like a lot of, they have a preparation, and also you sent a video how they prepare that.
BIOMED are prepared of fruits and whatever, so we can choose that part, but Belen can answer better.
Yes, sorry, I cannot turn on my camera because I'm in a place with very low signal. But yes, we investigated. We went to, well, we asked a lot of people, and they used one of the biological controls mostly used is called Trichoderma harzianum, and it's a fungus, and they used a preparation of different strains and well, different species of Trichoderma. And well, the problem was that firstly, they didn't have such a high specificity, and like specifically for Lasiodiplodia. So with our solution, we try to have like a better specificity and also reliability in comparison with the biological control that they use. That's Trichoderma harzianum.
Okay, great. Yeah, but just one quick question. So that's what you found with farmers, but when you did like a literature review, or did you find something else? Like online, like when you researched?
Oh well, yes. We also found other solutions with chitinases. So, for example, optimizing. Well, for example, we investigated about trichoderma, about ways of optimizing Trichoderma harzianum production of chitinases to combat fungi. Yeah, and the reason we did not go that road was well, firstly because there wasn't much information on that subject, and secondly because we found that there were also beneficial fungi that could be affected by this solution.
Okay, thank you.
I can go next. So your solution involves the so my first question is this a solution for plants before they get infected, or is this a treatment for infected plants? And then my next question is injecting trunks something that the farmers usually do? Is that something that they are used to doing?
This is a pathogen. Well, it's there is always in the plant, but it changed to be a problem when the plant is in stress conditions. So you can put this solution as a prevention treatment because we just are silencing two genes, and the effect is not 100% remove the lesion, reduce the lesion. So you can complement with a traditional pesticides that exist. So our real objective was reduce the cost of the farmers, because our product is not going to be the only and you need to apply. So that is not realistic. So the near objective was to reduce the cost that farmers invest to solution this problem. And that is yeah, that's the solution. So it's like a treatment, prevention treatment.
Gotcha. Thank you.
Okay, I can ask the question now. Hey everyone. So I have a question about even getting from what you just said. There is a minimum amount of RNI per plant that the eligible mine needs to be extinct, because you mentioned that can be death or decreased virulence. So there's a threshold there. And just a little follow up to that. Since you just said that it's already present in the plant, there are any possible ecological effects or populational effects if you extinct this pathogen to the plant? There are other consequences?
We don't found literature about that is beneficial to the plant. But with this technology, we don't believe that we are going to be able to extinct the pathogen, because the already solution doesn't exist, doesn't do that. So we're just reducing the lesion length, and our main objective is going to be to don't reduce the plant production because of this fungi, because of this pathogen. So well, we don't find scientific literature tell us if there is a beneficial that has this fungi on the plant. So yeah.
Thank you.
Are there commercially available RNAi based products for agricultural use? Is that something that is done currently?
Yes, we found one product that is already commercial in the market, but it's not available in our country. And when also in the Yogol, we present our solution to CEO of Syngenta, that they tell us that our solution is it has the potential to be applied in our country, because it doesn't break the policy that we have here, because in our country is don't allow it to do how to say it?
You can say
modificados.
OGM.
Modificados. So that was our first idea, to do an organismo modificado, but the policy in our country doesn't allow that. So we choose the idea of the exogenous application of double standard DNA. But yes, there is in the market.
Got it. Thank you.
Yeah, and to complement this answer, there is also, and there is not a specific biofungicide for this fungus in the market. So, and also this cost $1 billion of loss to the person, yeah, in the sector.
Okay, so I hope there's enough time for this question. I'll try to be brief. In your project, you claim that you have interviewed plenty of stakeholders, but I only saw that you only interviewed one farmer and one head of the farmer coalition. So how do you validate this part? Because as scientists, we also need to validate this part of the market. So what could be done next so you can further validate your business hypothesis and your solution?
Rosita.
Yeah, we only show one farmer, but this represents an average of 10 fundos in our coast. So it's a farmer who represents that. And also the same to the companies. We only show the main businessman, but he represents a average of 5 companies who sell this type of fungicides. So that's why we go to these persons. Yeah.
Well, perfect, time is over. So thank you, team. You can leave the room now. And judges, we're gonna wait for the other team to join the session. Thank you so much.
I'm still seeing Rosa online. There we go.
Only judges here now, right?
Hi, team. Please, can you change your name and add your team name? In this case, is BIOMED. So that will help us to know if you are in the right team. I'm gonna start sharing, now that we have team members from BIOMED, I'm gonna start sharing the promotional video.
Just checking, is the sound working?
No.
Esos eran mis cultivos de café luego de ser atacados por la roya. Lo perdí todo. Esos eran mis cultivos de café luego de ser atacados por la roya. Lo perdí todo. Las hojas no paraban de caerse. Ahora estaría en la quiebra de no ser por este grupo de jóvenes que vi en la televisión.
Somos BIOMED, un grupo de investigación formado con el fin de ayudar a combatir las plagas que afectan a los cultivos de importancia. Nuestro producto, Arroyer, es la solución contra la roya amarilla, un hongo que afecta al café generando pérdidas de hasta 108.17 toneladas, llegando a afectar al 5.88% de los productores, lo que significa una pérdida de más de 16 millones de jornadas. El cultivo del café es una actividad de suma importancia económica en el Perú, siendo la principal fuente de ingresos económicos para cientos de agricultores. Para crear nuestro producto, primero pensamos, ¿cómo afecta el hongo al café? El hongo Mileia bastatrix produce urodosporas. Estas ingresan por los estomas y parasitan las células vegetales. En este contexto, creamos Arroyer, una proteína modificada que evita el crecimiento y reparación del hongo y puede ser distribuida mediante aspersores y fumigación a las plantaciones de café, para así evitar pérdidas de este cultivo y apoyar a los productos nacionales. Arroyer es un producto eco-friendly, barato y de fácil aplicación, que beneficiará tanto la calidad de vida de los productores como el medio ambiente, ya que, a diferencia de productos pesticidas, este no tiene efectos nocivos en absoluto. Así que no lo piensas más y adquiere Arroyer. Contacta con nosotros y mantendremos tus hojas seguras.
Okay, time for question. Nana, then Ellen.
All right, team. Thank you for your very interesting presentation. I liked how you started out by asking, how are our leaves, right? I think you also have team members from different schools, so I appreciate the collaborative effort there. I saw that in your modeling. I think you had mentioned that your chimeric protein can kill off most of your spores in about 50 hours or so. I'm wondering how that statistic compares to current fungicides. Is it possible to model that compared to what's currently on the market?
Hi, please, can you repeat the question? A
Yeah,
little
so
bit
I saw
slowly,
it.
please.
So I saw in your modeling that you had mentioned that that your chimeric protein can kill off most of the spores in about 50 hours. I wonder if you were able to compare that with any commercially available fungicides. So is it better or worse, about the same?
Oh, okay, so when we analyze some papers about it, we found that the majority of analysis is based on fungal biomass, not exactly fungal spores, because it's a little bit difficult to quantify the germination. So we actually got the kinetic constants by analyzing some graphs on paper. So that's the way we did the model. We expect to test not only the experiment with our peptide, but also with commercial antifungal. So we can compare both graphics and analyze if our product is better, is worse, or is at the same level of the commercial biofungals.
Okay, nice. So if you eventually do this experiment in lab, you do plan on testing this against commercially available fungicides?
Yeah, exactly.
Alrighty, excellent. Thank you so much.
Hi, very nice presentation. I would like to ask about how you plan to get your peptides. It looks like you have his tags in your constructs, so I'm assuming you were planning to purify the proteins through a column. Proteins manufacture can be very, very expensive. And of course, in agriculture, you need a large quantity in order to treat hectares worth of land. What is the cost comparison for the production compared to chemical solutions for the same problem?
Can you hear me clearly? Okay, good morning with everyone. We actually have to test the protein in real life, with leaves, real leaves. And to clarify the amount of this protein we need for hectare, I don't know how to say that, area for a land, how much kilometers we are going to test actually. So we thought it was going to be a good idea use this instead of the fungicides on the market because it is a better option. Also, the fungicides not only are toxic, they are also expensive. So when we look after the prices on the materials we are going to need, I don't have actually the number, but we saw in a grand scale, it would be a better idea. I don't know if another member of the team have actually the number, the quantity. I can translate if you don't speak English quite well, but I think for a better answer to that question, we have to test our protein in real life to test effectivity, not only in the computational way, I mean, with results.
Thank you.
Hello, team. Congratulations on your project. I believe it's really important that you are working in this problem The coffee rust is ravaging our coffee production, and for a coffee snob like me, that's really good news. We need to protect our coffee from climate change and from all these pathogens. My question is, how can you test and compare the protein models, because the protein models that you got were from AlphaFold, and I'm concerned about the limitations of protein modeling regarding the actual behavior of complex molecules such as proteins. How would you validate and test the hypothesis that you'd made with the experimentation of these predicted structures?
Well, I want to start with a basis. For this kind of fungi, there is no more data, not much data. Instead of that, we decide to study another data with other kind of fungi, and taking that, we try to work into that. Also, I think it's complicated to work with a de novo modelation, and you can be certain about what the software can give you, what results can give you. That's why we made a mathematical modelation too. So that's like our backup. It's not just, well, the software has limitations. We can model the reaction of and the pattern of respond to our protein besides the fungi respond, no, or affinity to the plant. So it's not just that we are trusting the software. We need to be certain about the reaction, because in real life, there's not the same variables that we put on software. There are so many variables we're not taking, but that's why there's two sides, no? The dry lab that we're trying to, in this pandemic situation, there are so many studies use the softwares to figure it out how to work in real life. So there's the other part, the wet lab, where we're going to test what we think at first it was going to work. Jefferson, you have another answer to this question?
Yeah. Hi. The molecular docking is for chain gluten and for effect and effector.
Excuse me, is this an instructor or a team member?
Doctor. Yeah.
It's no, he's a team member.
Oh, okay. Sorry, his background said instructor.
Please, for present screen.
He wants to present some slides to show you.
Tengo inhabilitada la función de compartir pantalla.
Okay. Can you please give him the I don't know, authority?
Yeah, it's allowed.
Ya dice que estás permitido, ya. Thank you.
Yeah. Un moment, present. This is the folder of Steam. Yeah, it's a one poster for docking in protein with protein protein chimeric and glucan. Yeah, for doctor. For mainly, five models with the glucan and visualize interaction for chain glucans. Yeah, and affinity is it's -9 kilocal soda mol. No, sorry. Kilocal soda is a good affinity between glucan and protein chimet. Is one. Other is other other experiment of docking in anti-effectors with effector effector has 379. This is this interaction.
Yep.
One and yeah. Majo, ¿podría traducirme? It's complicated.
Yeah, sorry to interrupt you, just to be mindful of time because there are other questions. Just in one phrase,
Yes.
what type of experiments can you use with all that you have already told me? Validate these assumptions that you're telling me from dry lab. How can you validate them in wet lab? Which experiments? Mention a few.
¿Sí entendiste la pregunta, Jefferson?
No.
Ha preguntado cómo es que vas a validar los resultados del dry lab en el wet lab, de acuerdo a tus resultados.
Ya.
Dilo en español, yo se lo traduzco.
Mediante enfrentar la proteína generada, la proteína generada y purificada.
We're going to purify the chimeric protein and we're going to test it with,
con hongos germinados, con Hemilelia bastante germinadas.
Okay, we're going to cultivate the fungi and we're going to test our protein in the leaves with also, there are going to be already infected. So that's how we're going to test the activity.
I can go next. Congratulations on the presentation and the work team. I was wondering about the effectors that you're targeting with the project. What class of molecules are these effectors? And are these the only significant immune response from the plant?
Well, there is an actually immune response for the plant, but there's actually really slow. And the fungi, these fungi and other kind of fungi have the capacity to, from some way, kidnap the chitin oligomers, the oligomers of chitin that the plant with chitinases break in the fungi wall. So normally the plant with these oligomers of chitin can respond because it is like this this estímulo, no recuerdo cómo se dice estímulo, but this thing in the area can tell them, okay, I'm sick, I'm being attacked, I'm under attack. So these fungi have an effector called ECP6 that this molecule kidnap the oligomers of chitin, so it's hiding itself, and the plant says, okay, I think I'm under attack, but I'm not sure. So I can produce a big amount of chitinases. And we figured it out with the literature that we can plus or we can improve the effective of our product using a chimeric protein that binds into the cell wall and don't let the fungi grow or develop correctly. So in that way, it's gonna die. But the other way is to use an anti-effector. So we can target the ECP6 and tell him some way, you're not going to work now because you're gonna be busy with our peptide, and you're not gonna be able to catch the oligomers of chitin. So the plant is gonna have a better response, showing more receptors on the cell wall on the leaves. And that's the plus of the product, not just a chimeric protein, also an anti-effector.
Thank you.
I want to add some information. Actually, the name of the effector produced by the fungi Emilia bastatrix is called HESP379. But the effector that my partner mentioned, ECP6, was an effector present in another type of fungi, in the majority of them, in fact, like in Chlamydospora. But we based our searching with this effector, no? ECP6. But as my partner said, we want to target HESP379 with an anti-effector. Moreover, we want to develop a chimeric protein with the two parts that my partner mentioned. Other one called the cerebral one and MP3 domain, which binds to the fungi.
Thanks, Diego, for the explanation.
Oh, hey everyone. I'll go back a little bit to the modeling question. Great that you implemented a populational model. I really like this integration. And on that, where did you get the parameters of interest? I saw that some you estimated, but did you get from literature, or most of them you need to do some estimation? And with that in mind, there's a threshold on how much of the chimeric protein is needed to to lead the population to a level where they cannot affect anymore.
You're asking if this is gonna end with the problem?
In some way,
Something like that. But
there's a threshold of how much you need the protein.
Okay. But at first you mentioned some parameters, but I couldn't hear what does those parameters. I didn't understand that part of the question.
Yeah, if you estimated or if you get from literature.
Well, talking about the parameters of affinity, there's literature that says that a level lower than -9 is a better option to show there's affinity. You don't need such energy. It's going to happen naturally. So I think that's one answer to the question. The next question actually is really a bit complicated because we need to test it with real leaves. We need to test it if this is not going to affect the coffee grains, but we're using actually biobricks that everyone uses all the time. So there's no reason to have a bad reaction in population. Behind that, we think that if we can develop the idea, it's gonna be really tough for the fungi to adaptate so fast to these two targets. One, to his effector from the attack to the plant, and the other is to break or not let him develop correctly. So I think it's a pretty good idea to stop this rust, and it's actually simple to make another product, but not with this Fuji, with another kind of.
Additionally, we perform document essays so that know if the affinity will be high.
I think time is
and that's it.
over, guys. So I need to stop this session because yeah, we're on the 20 minutes now. Sorry for interruption. So team, you can leave the room and we're gonna start receive the other members. Thank you so much.
Thank you so much. Goodbye.
Okay, welcome team. We're gonna start by presenting your promotional video and then judges are gonna ask you the question. So the clock is gonna start right after the promotional video. Also, please add your team names in your nicknames in Zoom, so we know that everyone is from Fireblighters.
Their video is having problems with sound. I don't know if that's been corrected or not, but it wasn't their fault. So the subtitles were important for at least me to see. Yeah.
Sorry, what happened? What happened to the subtitles? Just checking.
I'm saying that I don't know if the issue has been corrected, but there was no sound in their video as it was posted on YouTube. So I was relying on the subtitles to know what was going on because I couldn't hear anyone talking. But Daniel or whoever's running the design league said it was not the team's fault and that we shouldn't judge them on that.
Okay, I'm gonna be sure that the next one also has subtitles.
I could hear something. Maybe it got fixed.
It's working now. So can I start?
Please.
Okay.
Los agricultores mexicanos necesitan ayuda. El tizón de fuego está acabando con el tejocote, fruta tradicional de la cultura mexicana, usada en el ponche, ofrendas, conserva e industrias alimentarias. Cada año se pierden más de 360 toneladas de tejocote por la bacteria Erwinia amylovora. A la plaga ocasionada por este microorganismo se le conoce como fuego bacteriano, el cual provoca que el sistema de defensa del tejocote se debilite, causando necrosis en los tejidos vegetales de la planta. Erwinia Erwinia amylobora primero invade el follaje y se reproduce. Luego pasa a los tejidos y órganos viejos y se manifiesta como si los tejidos estuvieran quemados y con un color amarillo ocre. Para controlar la enfermedad, tradicionalmente existen dos opciones. La primera es el uso de sprays a base de cobre que generan una acumulación en el suelo, dañando así el medio ambiente. Además, el hecho de ser efectivos tiene un efecto contraproducente para el crecimiento de la planta. La segunda opción son los antibióticos, cuyo uso continuo provoca que las bacterias generen resistencia a estos y cada vez se necesiten mayores cantidades para controlar la enfermedad. A pesar de que existen soluciones, estas representan desventajas. Por ello, se requiere de una solución adicional para controlar la propagación de Erwinia amylobora, ya que de no controlar la plaga de las cosechas, la economía de los agricultores mexicanos seguirá viéndose afectada. De esta forma, Fireblighters busca aportar una solución con bases éticas, normativas, sociales, medioambientales y económicas, usando genes y enzimas relacionados con los factores de virulencia de Erwinia amylobora, lo que ayudará a controlar la plaga para contrarrestar las consecuencias de los métodos tradicionalmente usados para el fuego bacteriano.
Perfect. We can start. I think first, who was the first judge to raise their hand?
I think Uriel.
Okay, go ahead.
Yeah, you're muted.
Oh, sorry. Thanks for that. Okay. Well, thanks, team, for your presentation. I really liked the video, and I learned something new. I didn't know tejocotes had this problem. I don't remember the last time I even had a tejocote, but yeah, that was interesting to learn. So I guess one of my questions is, so I understand that there is this problem of antimicrobial resistance, and this is why you chose the phages. But is there the possibility of the emergence of phage-resistant bacterial variants? Like if this pathogen eventually becomes resistant to phage, wouldn't that impede the treatment of your outcome? What you want to do, or do you think of possibility of that happening?
The phage that we are using is there is the lysogenic phage, and although it is known that some of these phages bacteria develop resistance to these lysogenic phages, ours is different because it is designed to kill the bacteria in some cases, or in other way, to let the bacteria live without a problem, only eliminating the pathogenic factors that it has. So we think, well, we developed this method so a bacteria couldn't have or wouldn't develop resistance to this phage, because it does not select like the bacteria. Well, we hope it doesn't select the bacteria for developing these resistances, as it doesn't kill them and it doesn't affect their growth. It is only supposed to cancel the pathogenic factors or kill them if it is in a high concentration in the plants.
Okay, thanks.
I can go next. Hello, team. Congratulations on your project. I did my graduate work on phages, so I really love to see it. My question is regarding the use of the lysogenic phage. So phages, and specifically lysogenic ones, are known agents of horizontal gene transfer in nature. They're known transfers of virulence genes. So my question is, why choose the lysogenic phage as a delivery system instead of using a lytic phage directly for biocontrol only? So just killing in this case, instead of silencing the virulence genes.
Because we wanted it, the phage to stay in the environment and to be able to, it's a long-term solution. So it's implemented once. It's hoped to be, it's aimed to be implemented once, and then it will it will be in the plan for a longer time. And yeah, we needed the lysogenic to control the plague and not to just kill the bacteria once and not to be and have to reapply them after some time. Like we wanted to be able to kill the bacteria or to reduce the pathogenicity in a controlled way.
Okay, thank you.
All right, thank you for sharing this project with us. I enjoyed how you tied it so closely to the culture of your region, and you've even included as a song about it. So I think you talked about this idea of how climate change impacts Erwinia infections, right? And you stated that strong winds and rains can cause Erwinia to become more prone to fire blight. Can you tell us a little more about this relationship between climate change and Erwinia?
Well, as we speak to the farmers in the area, we discovered that there are certain times or seasons where the plant is more prone to be infected. And it is because of rains that it one plant infects another's. So all these changes in climate make it more prone that this bacteria is more infectious to other plants.
Oh, interesting. So the more rain actually propagates the infection.
Yes.
Okay, interesting. All right, thank you.
Is it Uriel or Ellen that is next?
I'll ask a really quick question. You had a section on biosafety and biosecurity. Can you define the difference between biosafety and biosecurity?
Yes, the biosecurity is related with the human health. It ensures that the product that you are developing has no risk to the human health. The biosafety is more related with that you have to analyze and study what are the possible impacts of your product, of your biosystem control, for example, for the environment. And how could you increase these risks and what measures you need to take?
Thank you.
Hi, so I actually forgot to lower the hand, but now that I'm here, I think I have another question. Just a quick one. So you mentioned earlier that you want the phages to stay in the environment long term. So I guess my question is, you said in your wiki that the phages are specific to their targets, but if you're intended to leave them long term in the environment, is there the possibility that they can impact other microbial communities that are present in the soil or in the plant? Even though you say they're specific, but could there be the possibility that they start attacking other types of bacteria?
When we designed the biosecurity and biosafety strategy of this project or this biosystem control, we take into consideration two aspects of biosecurity and biosafety. That is when we create biosystem controls and when we use fakes. For this is like essential, according with the scientific bibliography that we consult, that the fakes need to be specific, and also, yes, we want to be to the fakes in long term, but also another recommendations that we found or we investigate is first the deletion of essential replication genes of the fakes, just to not impact too much the environment, and also the main deletions, virulence factors of the fakes, that they are not the genes that we are inserting. And also we take into consideration another aspect, that the genes that we are inserting in fakes are specific for Erwinia amylovora. And we did analysis on what bacteria are affected for the genes that we are using. So there are the biosafety aspects that we take into consideration when we were designing the fakes.
Great, thank you.
Hello, team. Congratulations for your presentation. Related to human practices, I am convinced that your work facing a local problem. My question is, what would happen if the Jocote farmers have resistance to use these new biotechnological products? What strategy are you working with them to explain the benefits of your product, and how do you involve them in this process of your solution?
Well, one aspect that we considered in the project to approach to the farmers was that to get an intermediate organization. In this case is CESABEP. The name is about, it's an organization that also controls some plagues and of some fields in the state of Puebla. So this organization has an approach, a close approach with that farmers. So we consider to first talk with this organization, and then that this organization help us to have a contact with farmers.
Okay, thanks.
Hello, team. Congratulations on your proposal. The Jocote is really important, especially with the Christmas season coming closer. And I have a specific question regarding entrepreneurship, because you want to, and you're planning to have an entrepreneurial recognition. So my question is this problem big or important enough to design a really expensive synthetic biology solution? Because you have mentioned that there is a lot of factors, but you have not mentioned the economic impact of this problem. So how it can be implemented in economical terms, what is the economical feasibility, and if this solution can be expanded to other similar problems?
Yeah, thanks for your question, Alejandro. In this part, we have into consideration the global market, because it's in growing. So we can take this project and scale to other fields like rosaceas or other rosaceas, and impact in the state of Puebla and the local local zone, and then take this project to scale in a national scale, and then grow in from a startup to a enterprise.
Thank you.
Hi, team. It's me again. My next question is about antibiotic resistance. You mentioned that one of the reasons for choosing phages is that antibiotic resistance in Erwinia is an important consideration. I was wondering if that's already observed in the field. Do you already see antibiotic resistant Erwinia strains? And if so, are these genes well characterized? And I'm asking specifically for safety questions because if the genes are in specific ways organized into the genome, phages are a great candidate for transferring them. So is antibiotic resistance something that is already observed, and is it characterized?
I think unfortunately we're like the first ones to be observing this type of plague in Armenia. Well, no, I don't mean the first one, but it's just developing. So I don't think there's a characterization of this antibiotic resistance. But yeah, as you mentioned, it should be. And yeah, we are developing the project more along the biosafety way. Yeah, because it's important because of the things you mentioned.
Thank you.
Hi again, team. You mentioned in your abstract that the ENT90 phage will be hosted in P agglomerons. Were you going to apply the P agglomerons in the field? And if so, isn't that a genetically modified organism? And isn't that against the laws in your country?
Mexican normative is not very specific with genetic organisms. It's not very developed in the Mexican normative about this topic. It's more focused, it's more centralized, talking about plants which are transgenic. That's a full topic, a whole topic in Mexico, but it's not really clear about the use of eggs and also the use of modified genetic bacteria. So that's the reason also why we create like a law proposal to try to begin to make the normative more bigger, more specific. We know that if we want to apply this product, the normative that we have to comply is related with some the general law health, and also we have to make requests or permissions to the CBOHEM and for COFEPRIS. But I don't think that is violating the normative, and this is also related because the Mexican normative is not to develop it with this topic. It's more focalized in transgenic plants.
And do you have an equivalent of an EPA, an environmental protection agency, that you have to deal with?
Also, well, I would like to add that as the phage is not modifying any genes in Pantoea, we expect that, well, it doesn't count as a genetic modified organism because the genes that we selected for modifications in the case of Erwinia millivora are very specific genes. They are pathogenic genes that are only expected to be seen in this specific Erwinia bacteria. We do have some organisms that do protect the environment. That would be the I don't remember the name, but yes, we have environmental protection organism. I don't. What was your question again? Yes.
So that the phage, if you're going to put this second bacteria in and actually apply it, so you're saying an infected bacteria doesn't count as a genetically modified bacteria. So it never becomes lysogenous in P agglomerans?
Well, yes, it does. But the proteins that are expressed only affect
Okay, sorry, I'll bow out and let someone else ask a question.
All right. I think Beatrice got me interested in this idea of antibacterial resistance. So you guys had mentioned this idea of copper sprays that are currently being used, right, to target Erwinia. What is the mechanism of action of copper to Erwinia?
Well, as many bacteria have this effect that it interacts with the membrane because of the charge. And yeah, as far as I understand, it makes the membrane break. So yeah, with there's a wall and the membrane, so it's just like a cell rupture as far as we have investigated.
Okay, so it sounds like this would affect a lot of microbes, not just Erwinia, right? All right, thank you. So so there's a lack of specificity there. All right, thank you.
Okay, if we don't have more questions, like we have 10 seconds, so I think we're done with the questions now. Thank you, team. You can leave the room.
Thank you very much.
Thank you.
Exactly.
So we have like almost 5 extra minutes before the other team. So we can take this as a break. We're gonna have this room, and yeah, we're gonna start observing the waiting room for the next team and the last one.
Probably a good time for a bio break.
Yes.
I'm gonna go eat a little bit of something very quickly. Be right back.
Hi team, please add your team's name in your Zoom's nickname so we can identify you and know that you're in the right Zoom. And you have some time to invite your other team members to join the session. Perfect. Welcome, team. So we're gonna present your promotional video, and right after that, the judges are gonna start asking you questions. You're gonna have 20 minutes, so we can start.
De maíz amarillo y de maíz blanco se hizo su carne. De masa de maíz se hicieron los brazos y las piernas del hombre. Únicamente masa de maíz entró en la carne de nuestros padres. Popol Vuh. Desde la época prehispánica, el maíz ha sido un elemento fundamental de la cultura mesoamericana, así como un componente importante dentro de su dieta alimenticia. Para muchos, clave en la concepción global del universo. Un cultivo representativo de México con importancia económica, social y cultural que forma parte de la vida de los habitantes de la región. Pero sobre este importante cultivo se cierne una amenaza, el gusano cogollero, también llamado oruga militar, que causa daños serios al consumir las hojas, inflorescencias sin desarrollar, daño directo a la mazorca e incluso los tallos. Una plaga que si no se controla puede acabar hasta con el 60% de la producción de este y otros cultivos, y hasta con el 100% como trozador. Y esto no es todo. Tras de sí, en un esfuerzo por erradicarlos, los agricultores hacen uso de múltiples químicos peligrosos, dejando así graves pérdidas para los agricultores y sus familias, ya no solo económicas, sino también de salud. En adición a ello, los ecosistemas son también afectados por estos tratamientos químicos. En una búsqueda por resolver este problema encontramos que la pimienta negra tiene una sustancia llamada piperina, que funciona como insecticida del gusano cogollero. Por ello, hemos diseñado con herramientas de biología sintética a Piper, un biopesticida hecho con una sustancia similar a la de la pimienta llamada piperamida, cuya información esencial introduciremos en una levadura. Sí, la misma con la que se hace la cerveza, solo que en vez de crear cerveza, creará nuestro biopesticida. Un biopesticida que ataca al gusano cogollero, aumentando la productividad, que es amigable con el medio ambiente, no tiene repercusión en la salud de los agricultores y es más económico que un pesticida convencional, porque los agricultores son el pilar de nuestra civilización. Somos Synthetic Biobots.
Okay, so we can start with Uriel. I think who raised your hand first? Yeah.
Yeah, I think it was me. Yeah. Great. Thank you guys for your presentation. I liked your wiki. I like the video. I don't think we've seen any stop motion type videos here at the Agile Design Week yet, so that was good. Okay, so I want to start with a question. So I've actually done work with biocontrol against this pest, and I know that there are other types of biopesticides that can be used to kill this pest, like cryproteins from Bacillus thuringiensis. Did you look at other types of biocontrol agents, or when you did your design process, why did you ultimately decide to use this compounds from the that you're trying to use? Yeah.
Okay, well, in this case, we were based on a paper that we insert in our references because we found it to be like very, very useful for this specific pest. And We did look for another pesticides, but the thing here is that we found that the thing with pyramid is that it looks that it's very efficient, and also as is very safe for human health, as you can see that, well, we actually eat it in black pepper, and we find it to be very a mix of a very safe compound, and also we're based on a paper that says that it's very damageful for the fall armyworm. So that's why we were like a bit of biased to use this instead of any other pesticide or any other compound.
Okay, thank you.
I can go next. Hello, team. Congratulations on your work. I really appreciate how you incorporated ancestrality and the value of the crop and the culture to your region. That was really nice to see. And also, congratulations on the metabolic engineering work. This is very similar to some projects we do here at Ginkgo, so it felt familiar, so that was nice. And my question is, you show your metabolic model of all the reactions you're planning to insert. And I was wondering if the model identified bottlenecks, and if so, what is the hardest part of the pathway? What do you anticipate will be an issue on this pathway?
Okay, I could say that maybe one of the most difficult parts is to really they have actual experimental data about if the enzymes actually work, because, well, as you saw, we were based on molecular docking, and therefore we are not 100% sure that it's going to use the model of enzyme of substrate is going to catalyze the reaction. So actually, that's like the biggest problems. And we did have some concerns about that, specifically about some of the proteins. Some of them are very sure that are going to work, but for example, the step number 10, I remember that has like a bit of a slower energy to bind to the molecule. So there are some difficulties about the molecular docking process and to really know if they're going to work.
Thank you.
All right, I want to echo what Beatrice said about integrating your culture into this. I think that's very important, and also what Uriel said about the stop motion animation, not just the video itself, but also your pictures, and then your all the figurines and characters you made out of clay. That was quite impressive. So I'm interested in learning more about how you decided to use the host yeast. Many other teams seem to rely on E. coli. How do you go about choosing this host?
Yes, the main reason we choose to use this yeast instead of, for example, E. coli is because we want to express several proteins. And as we said, the best expression pattern we found was this temporal program. When we express some proteins at the beginning, like in this constitutive way, and some proteins are expressed in an inducible way. In order to do this, we found that the best way not to have a lot of transcript units, like 10, but just only 3, it was to use this polycistronic system. But the proteins we are using are not, the genetic code is not the same for plants, for bacteria. So the best way we found to do this was to implement this in another eukaryote system like the yeast. And we found a way that we could implement this polycistronic-like expression system in an eukaryotic expression system that is Saccharomyces.
All right, thank you.
I also want to add that as we are using all of our proteins are from eukaryotic organism, a eukaryotic organism, a plant. Well, it's more reliable to use an eukaryotic system rather than a bacteria. So that's another important reason.
All right, thank you.
Hello, team. Congratulations on your promotional video. It's really informational and it's really fun to watch. And corn is really an important part of our culture, like thousands of dishes depend on corn. So I'm glad that you're working in this project. Following the previous questions, I actually had some questions regarding the chassis that you are choosing, which is yeast. So with what have you already said, considering these proteins and these polycistronic constructs that you built, are there other considerations that you took for the proteins to be expressed in yeast, such as, let's say, folding, post-translational modifications? Because we know that some bacteria also have these polycistronic systems. So I wonder, like, why did you choose yeast? And also, excuse me, does piperamide biosynthesis affect yeast growth, like cause any kind of inhibition that would impact your bioproduction?
Okay. In the case of why, another reason why we choose to use yeast was the size of the genes we were going to transform the bacteria or the yeast in this case with. For example, we use some yeast artificial chromosomes that allows us to transform these yeast with a large amount of DNA with a big construct. So that's another reason why we choose to use yeast instead of other bacteria, for example.
And can you answer the part of the growth inhibition by piperamide in yeast? Is there any information that you consider or available in the literature?
Well, we didn't actually consider the inhibition by drug production. No, the answer for that question is no. And well, yeah, that's it.
How would you test this, just to end this part of the question?
We can do like this exercise where we prove different concentrations of the active compound we're using, like to search for the lethal dose 50, maybe in our expression system, like how much concentration of this compound the yeast can produce before it affects its own growth, for example.
Great, thanks.
Hi, I again echo everybody else. That promotional video was an excellent science communication example. And congratulations, a lot of hard work went into it. My question again, and I think we're all asking questions about the same thing, because to wrangle 11 different genes all at the same time in a pathway is extraordinarily difficult. My question is that you've addressed a temporal problem, but what about the amounts of each enzyme in the pathway? Chris Voigt gave a really great lecture at a synthetic biology meeting about trying to do a pathway with 16 different genes, but they all had to be expressed within very small quantity limits. And he said it was a problem in 16-dimensional space. So how will you address the problem of the amounts of each of these enzymes in the pathway? You did give some information in the pepper plant, I believe, transcriptional analysis. So what will you do next to try and look at this problem if you end up not getting the result that you want on the first try?
Okay, One of the options that we have to address this would be to change, for example, the order of expression of proteins, which is somewhat malleable because we can change it in the construct. For example, if we found that some enzyme needs to be in a greater amount than the others, we can test it, and we can test if that's a better approach. And yeah, it's indeed a very challenging task because it's very difficult to really know what's the most optimal way to do it. But one of my proposals is to change the order, and for example, we can express and polymeric enzymes first, rather than because of the temporal program. So we could express first the biggest or the most amino acid consuming enzymes first, and the polymeric, because we expect them to be using the most amount of resources. So that could be one way to do it.
Thank you.
Hey everyone, like you say in Portuguese, I'll be raining on the wet, because I would say again, amazing presentation. It was a great example of science communication. Really loved it. I love stop motion. So, but I'm going to a little bit different path. It's a quick question. Do you think there are any possible ecological effects of this solution that you're proposing, especially thinking you're working a solution for insects? It's possible to affect other species of insects, especially if we think they are crucial to the ecosystems?
It is possible that it can affect other species, for example, of lepidopters, because it is not specific for this species. It is also important because Spodoptera frugiperda is not the only species, for example, of Lepidoptera that affects these crops. So it can be useful against other plagues. But the fact that it is less harmful with the environment, it is used in a similar way that other pesticides, but its impact on the environment is lower. So yes, it is possible, but its effect is much lower than other pesticides.
Okay, thank you.
I can go next. Does this molecule have taste? Are you seasoning the corn when you apply it to the plant?
I'm not sure. It gives flavor. I mean, like, it's present in the black pepper, but in the period it is going to be applied to the corn is like before the corn grows, actually. So it's going to be like in this part of the plant that is called in Spanish, in Mexico, it's called cogollo, that it's part of the leaves. So there is where the fall armyworm attacks the plant mainly. So the main period when it's going to be applied, it's before actually the crop actually grows its fruit.
Got it. Thank you. And just adding to how you're going to engineer that, come to Ginkgo. Let's work together. We can do this.
Thank you very much.
Do I have time for a question still? 5 minutes.
Yes.
Yeah.
5 minutes. Yeah. Well. 4.
Okay. Okay. So my question is, so imagine you managed to produce this, compound. What would be the process of making a proof of concept for for what you want to do? What sort of experiments would you do to test if your compound works? Because from what I saw on your wiki, you cited a paper that shows that piperine has an effect on the hatching of the larvae, but would it only work on hatching larvae, or would it work on full-grown bloodworms? So, like, if you use this on full-grown, insects, would that work? And what experiments would you do to test it?
Okay, I think one of the papers, we cited one paper, but there are some other papers that say that this compound affects not only the hatching of the larvae, of the eggs, but also the larvae that affects the crop. So yes, it can kill also the larvae, not only impair the hatching. So it has these effects too. And we will prove it like it could be some experiments like this little doses 50 to know the amount of the concentration of the compound necessary, like to kill or impair the hatching of the eggs. Maybe in a similar way, some of these papers, I say there have been some papers, not just the only one we cited, that have made these experiments to prove the effect of the compound on this species.
Okay, so you do some sort of bioassays. Okay, thank you.
Okay, with the time remaining, I'll try to be super quick. Why should you go through the trouble of producing piperamide with Symbio instead of extracting piperine from black pepper? How is this more economically feasible or attractive than just extracting it from what's already existing on the market?
Okay, one of the main reasons we choose to work with this problem with Symbio is because of the area we're working with. We're working in Mexico, in the states of Michoacán, Jalisco, Sinaloa, and in this region, for example, the crop of the black pepper is not affordable. Why? Because we make a model of ecological niche model to know if we will be able to crop this black pepper fruit in these regions in order to extract this compound. And we found that it was not suitable to grow this crop in these regions. So we thought that in order to avoid like these travel expenses and all the infrastructure necessary, like to produce the fruit in one place and extract it in another and then transport it, we thought that it will be something possible to produce this compound using synthetic biology.
Do you have any figures or economic numbers to support that assumption?
No, in this case.
If we have a 2 seconds, very quick question. Great work with your material for people with visual impairment. Did you apply an activity with this material? I really like this idea.
Sorry, I didn't listen clearly.
If you applied any activity with the visual impairment material that you created, because it's a great idea. I really like it.
I didn't understand the question either. What do you mean apply material?
If they use that with folks?
Yeah, if you actually applied an activity with this, or if you just create.
They use it in real life, yeah.
Yeah.
Yeah, we are just based on papers that we found and information and research, not actual laboratory work or real work.
Okay, I think time is over, so we can finish the session here. And thank you, team. Great job, and it's time to leave the room.
Thank you.
Thank you. Goodbye.
Thank you, team. Bye.
So I got something from Daniel saying that there's a different room